Which of the alleles are present in any particular individual, the locus for all those alleles would be same on the chromosome. If this is the chromosome, there are 3 loci, the first one and second one, third one. Let's say a particular gene has 6 alleles. An individual can have only 2 alleles. One particular individual might have allele number 2 and 3. Both of them will be in the same allele. Let's say this is the location. Location of genes on the chromosome is called a locus. That all of you know. Location of the gene on the chromosome is locus. Number of alleles for a particular gene is dependent on what? In an individual, it is dependent on the ploidy of the individual. If an individual is diploid, every gene except the genes on the sex chromosomes will be having how many alleles in a diploid individual? 2 alleles. So alleles will always be represented on the corresponding locus of homologous chromosomes. Homologous chromosomes, chromosome number 1, chromosome number 1. Every region, every locus on both these chromosomes, the corresponding locus will have the same gene. It is the same gene that is represented in the respective locus. But different chromosomes in the homologous pair can have different alleles. So if gene number A, B, C, D, E is present in one of the homologous chromosomes on locus 1, 2, 3, 4, 5, on its homologous chromosome also, on locus 1, 2, 3, 4, 5, you will still have genes A, B, C, D, E. You will not have change. If you overlap those chromosomes, the same genes will be overlapping each other. Alleles can be different. This chromosome can have a dominant allele, this chromosome can have a recessive allele. That might be different. So multiple alleles are present where? At the same locus on the chromosome. Alright? Which body of the government of India regulates genetically modified research, genetically modified organisms research and safety of introducing GM organisms for public services? It is just a statistic, just a data point. Biosafety Committee, Indian Council of Agricultural Research, Genetic Engineering Approval Committee, Research Committee on Genetic Manipulation. Option 3, Genetic Engineering Approval Committee, GEAC. Okay. What does it oversee? Safety regulations, approvals and research related to genetically modified organisms in India. Alright. Others are not associated with ICAR, Indian Council of Agricultural Research. That is the agency or that is the organization which approves new varieties of crops. That is the organization which approves new varieties of crops. Alright. In BT cotton, the BT toxin present in plant tissue as a protoxin is converted into active toxin due to alkaline pH of the insect gut, acidic pH of the insect gut, action of gut microorganisms, presence of conversion factors in the insect gut. The insect gut is alkaline or acidic? Alkaline. Alkalinity of the insect gut will activate the BT toxin. Alright? BT protoxin becomes the active toxin in the alkalinity. Now, another logic given of the safety of BT crops, BT food products for humans is that the activation of the BT toxin is in an alkaline environment. BT toxin, the protoxin, whatever is present in the cells of the genetically modified crops, gets damaged in the acidic environment of the stomach. Alright. So the acidic environment of the stomach damaging the protoxin, leading to the Bt toxin becoming inactive in the human gut. Therefore, the Bt toxin cannot be activated, and therefore it is not to be worried about, is one of the logic. But it is such a microtoxin, such a small level, its effects cannot be studied so conclusively on human tissue in the actual physiological conditions. The debate continues. The crops engineered for glyphosate resistance are resistant to fungi, bacteria, insects, herbicides. See what happens in what are the insecticides trying to do? Insecticides or herbicides? The metabolism in the plants will be disrupted by herbicides. Now certain herbicides, the chemicals in certain herbicides will act on certain metabolic pathways in some weeds. So herbicides, chemical herbicides should be designed in such a way that a particular chemical disrupts the metabolism of a particular plant species. That metabolic pathway should not be a prominent one in the crop. Otherwise, what happens? You are disrupting a metabolic pathway in a weed and destroying the weed. If the crop plant also has the same metabolic pathway, it will destroy the crops also. So if you want to use a particular herbicide or a particular chemical on weeds, you can develop plants with resistance to that particular chemical, thereby protecting the metabolic pathway which this particular chemical will disrupt. Alright, so glyphosate resistance allows plants to survive glyphosate based herbicides. So those plants which have the resistance will not be affected by this. Those plants which do not have the resistance will be affected by the chemical, and it gets destroyed. DNA is not present in chloroplast, ribosomes, nucleus, mitochondria. Where do you not find DNA? Where do you not find DNA? Mitochondria, no? Mitochondria has its own DNA. Chloroplast has its own DNA. Ribosomes, what do they have? What do ribosomes have? What do ribosomes have? Do ribosomes have nucleic acids? Do ribosomes have nucleic acids? Do they have nucleic acids or not? What are ribosomes made up of? Proteins. Only proteins. Name the 3 types of RNA. What is rRNA? Do ribosomes contain nucleic acids? Do ribosomes contain nucleic acids? What nucleic acids do they contain? RNA. They have RNA. Correct, no? So, see, trying to remember and recall and trying to arrive at answers, arrive at solution, logically, different. You have to think, no? Do they contain nucleic acids? What nucleic acids are there in the body? DNA and RNA. Ribosomes do not contain DNA. If the question is, do they contain nucleic acids, then is there a possibility that they have RNA? If you are not able to suddenly think about it, think of different types of RNA. Ribosomes do have nucleic acids, it is RNA. Extra chromosomal nucleic acids are found in dash. Give me, name the organelles or places in the cell where you find extra chromosomal nucleic acids. I'll give you options. Cytoplasm, ribosomes, or extra chromosomal nucleic acids are absent in cytoplasm, ribosomes, Mitochondria, plastids. Extra chromosomal nucleic acids are absent in. Huh? What? Cytoplasm. Okay. Huh? Ribosomes. Now only we spoke about RNA being present in ribosomes. I am talking about extra chromosomal nucleic acids. So it includes both DNA and RNA. Huh? Plastids. In the same question we saw chloroplast has its own DNA. Chloroplast is also a plastid, no? Huh? Mitochondria. Mitochondria also has its own DNA. Huh? Mitochondria has can have its own DNA also, and it can have its own ribosomal machinery also. Protein synthesizing machinery also it can have. Then only option left is cytoplasm. Where is tRNA? Where do you find tRNA and mRNA? mRNA and tRNA are found where? In the cytoplasm. So the answer is none of the above. I did not give you a multiple choice question. I asked you of these four, which one does not have extra chromosomal nucleic acid? You can say none of the above. Alright? Okay. Which of the following enhances or induces fusion of protoplast? What induces protoplasm fusion? Sodium chloride and potassium chloride, polyethylene glycol and sodium nitrate, indole acetic acid and kinetin, indole acetic acid and gibberellins. Protoplast fusion by reducing surface tension and membrane fusion is facilitated by, one you should definitely be knowing, it is directly out of NCERT. Protoplast fusion is brought about by polyethylene glycol. Polyethylene glycol and sodium nitrate. Indole acetic acid and kinetin. Kinetin is what? Kinetin is cytokinin. It is the same thing. Both are growth promoters. Indole acetic acid and gibberellin, they are also growth promoters. They do not bring about protoplast fusion. Alright? And sodium chloride and potassium chloride are not used at all in protoplast fusion. UN Conference of Parties on Climate Change in the year 2011 was held in South Africa. One of these UN Conference of Parties on Climate Change was held in Kyoto, Japan, where we got the Kyoto Protocol. Vertical distribution of different species occupying different levels in a biotic community is known as vertical distribution. Stratification. Strata means layers. Layer is always one above the other. Layer is always one above the other. Those are layers. Stratification. What is divergence? Divergence, where do we find divergence as a concept? Evolution. Divergent evolution. Okay. Zonation. Zones. Zonation is the formation of zones. Zones are always horizontal. When you are looking at distribution horizontally, you are looking at zones. When you are looking at distribution vertically, you are looking at strata. You are looking at strata. Stratification is layers. Formation of layers. Pyramid. Pyramid is a depiction of different components in the food chain or in the ecosystem. How different things move from one trophic level to another trophic level. In which of the following both pairs have correct combinations? In situ conservation, national park. Is it right? In situ conservation, national park is correct. Ex situ conservation, botanical garden. Both are correct. We need to look at all the other options. In situ conservation, cryopreservation. Cryopreservation is not in situ. Cryopreservation is what? Preservation in a frozen form. You freeze some tissue. You freeze some tissue by using what? Liquid nitrogen. You use liquid nitrogen and freeze it, and that is cryopreservation. Wildlife sanctuary, ex situ conservation, is it right? Wildlife sanctuary, ex situ conservation. Sanctuary is not an ex situ conservation. Zoo is ex situ. In situ conservation, seed bank, that is also wrong. Ex situ conservation, national park, that is also wrong, reverse. In situ conservation, tissue culture, ex situ conservation, sacred groves. Again, reverse, if you reverse it, they are right. Secondary succession takes place in or on bare rock. What is bare rock succession? Primary succession. Degraded forest. Is that correct? Secondary succession, degraded forest. What succession will happen in Los Angeles after the forest fire? Is secondary succession. Thousands of animals died. Very painful sight to watch that. Newly created pond, is it primary or secondary succession? Primary succession. Newly cooled lava, primary succession again, because lava would completely eliminate whatever is existing already on the surface. Burning will still retain the texture of the soil and all that. The organisms might still remain. Lava will completely eliminate everything. You should start new. The mass of living material at a trophic level at a particular time is called gross primary productivity, standing state, net primary productivity, standing crop. What do you call the mass of living material at a trophic level at a particular time? Standing crop. You call it as standing crop. Standing state is a reference to inorganic nutrients in an ecosystem. The living organisms in an ecosystem is standing crop. Inorganic nutrients is standing state. Net primary productivity is what? Or gross primary productivity? Primary productivity is the amount of biomass produced by producers. Amount of biomass produced by producers is primary productivity. Gross primary productivity is what did they produce before using for their own consumption. That is, plants produce biomass, let's say carbohydrates, glucose and all that. They use a part of it for their own respiration also, for their energy needs also. Before using it is gross primary productivity. After using it is, whatever is left out is net primary productivity. Your gross salary and net salary. What is the gross salary? What is on paper? Net salary is what comes to your hand after deductions, after TDS, PF, professional tax. Yes, if you have any loans and if you have linked your salary, all the EMIs, everything cut, and what you get in hand is net salary. Gross salary will be big. Net salary will be enough for 10 days. If you don't manage your finances well. Alright? Something that I have not followed, but I teach everybody and I am ensuring that my wife is applying it, is whatever is your income, you should consider only 50% of it as your disposable income. For all your expenses, living cost, everything should be within that 50%. 50% of your income should just go for investment. You should invest 50% of your income. Before it comes into your hand, you should have made arrangements for 50% of your income to go into investments, which you will not touch. So if you are earning 2 lakh rupees a month, 1 lakh rupees should by default go into investments. If you are earning 50,000 rupees a month, 25,000 goes into investment. You only have 25,000 to do your living. If you consider 2 lakh as your income for expenditure, when you are going somewhere, you will book a star hotel. You will spend on luxury dinners or fine dining. If you cut it down by 50%, push it into investment, and you are living only with 50% of your income. You should manage with that. You should not take loans to deal with your living expenses. You should manage your living expenses within 50% of your income. As your income keeps increasing, your lifestyle increases. You cannot use your entire income for your lifestyle. Your lifestyle should be within 50% of your income. No matter how much you earn, 50% should go into investment. If you do this, you will become wealthy. My wife is more wealthy than I am. All right. Because I have various other ways of spending money, not for myself. A lot of things I do which requires a lot of money. So, all right. Anyways, net primary productivity, gross primary productivity. Right. In an ecosystem, the rate of production of organic matter during photosynthesis is termed what? During photosynthesis, what do you call it? We have discussed this already. You have to tell me the answer. What do we call this? Gross primary productivity. Gross primary productivity. Secondary productivity is what gets transferred to the consumers. All right. Net productivity is what is available at each trophic level, not an individual organism. At every trophic level, whatever is the biomass, energy in terms of biomass available, that is net productivity. Which of the following characteristics is mainly responsible for diversification of insects on land? What is responsible for diversification of insects? That means what gives them the advantage of survival in a variety of conditions. Why do a particular species or a particular phylum gets or a particular class gets more diversified? You find it everywhere. It has developed the ability to survive in varying conditions. So what has given the insects that kind of an advantage is what you have to see. Segmentation, does it provide any special advantage? No. Bilateral symmetry, exoskeleton, eyes. Exoskeleton is what gives them advantage. So exoskeleton is the choice there. Which of the following endoparasites of humans show viviparity? Viviparity. Viviparity means what? Viviparity is production, giving birth to young ones. Should it be a fully functional form, or even if it gives birth to larvae, can we consider it as viviparity? Let's say instead of cockroaches laying eggs, if they were giving birth to larvae, should we consider it as viviparous? Kangaroo does not give birth to a fully developed baby. It gives birth to a partially developed baby, and the baby's development, the fetus's development completes in its pouch. So is it inside or outside now? It is at the door. Neither fully inside nor fully outside. Alright. So should we consider it what? Viviparous? It is viviparous. So in this Trichinella species, Trichinella spiralis produces live larvae, okay, that migrate to the host tissue. The others, Ankylostoma duodenale. Ankylostoma duodenale is what? Ankylostoma is hookworm. Ankylostoma is hookworm. Enterobius vermicularis, Ascaris lumbricoides. Ascaris lumbricoides is our noodles, roundworm. They are all oviparous. Which of the following represents the correct combination without any exception? No exception. Mammary gland, hair, pinnae. No exception means all the organisms belonging to that particular class shows it. No exceptions. Alright. Mammary gland, hair, pinnae. Is it present? Is all three present in all mammals? Yes, no exceptions. All mammals have mammary glands, hair and pinnae. Chondrichthysis. Chondrichthysis, cartilaginous fishes. Ventral, mouth and placoid scales. Do all the cartilaginous fishes have ventral, mouth and placoid scales? Can you think of any exception? You have to look for exceptions. Sucking mouth, no jaws are characteristic of Cyclostomata. Feathers, moist glandular skin are the characteristics of aves. All four are characteristics of the classes that are represented by it. Mammals definitely have mammary glands, hair and pinnae. The others, we have to look for exceptions. Let's find out exceptions. I am not able to recall the exceptions right now. Alright? Can you think of any exceptions for these characters? Do all birds have feathers? That we can think of an exception. Penguins don't have feathers. So featherless birds are known. Sucking mouth, no jaws, cyclostomata. Okay, we'll figure out. Which of the following animals is not viviparous? Flying fox. Flying fox is what? Flying fox is a bat. Elephant is viviparous. Platypus, whale. All of the four have mammals. All the four are mammals. Platypus is oviparous. It's the egg-laying mammal. Erythropoiesis starts in kidney, liver, spleen, red bone marrow. Now this is an incomplete question. Erythropoiesis is the formation of red blood cells. Erythropoiesis is the production of red blood cells. Where does it start? Blood formation. Now we can look at it in two ways. In adults, see, erythropoiesis, do we consider it as RBC production or blood production? RBC production. WBC production is called leukopoiesis. Alright? Platelet production is called megakaryogenesis. Megakaryopoiesis, you can say. Now, erythropoiesis happens in the bone marrow, no doubt. If you say begins in, what do we have to consider? Do we have to look at it as in adults or in fetal life? Or beginning of blood formation in human beings? If we have to look at it that way, beginning of blood formation in human beings, what should we consider as the right choice? Liver. Liver is the first organ which starts producing RBCs in the intrauterine life. But in adult life, red bone marrow is the one which produces RBCs. So, what do we choose? The answer given is liver in answer key. What is the answer key telling? Liver? Very good then. Liver. Alright? Because starts in is to be considered as beginning of blood RBC production in human beings. So in the fetal life, it is liver followed by spleen, then bone marrow. What retains the RBC producing ability? Liver retains the RBC producing ability, spleen loses it permanently by the time the child is born. If bone marrow failure happens, liver can restart RBC production, spleen cannot. Okay. What is the role of kidney in RBC production? Role of kidney? Erythropoietin hormone. Terga, sterna and pleura of the cockroach body are joined by. Terga, sterna and pleura of the cockroach body are joined by cementing glue, muscular tissue, arthrodial membrane, cartilage. The connecting junction has arthrodial membrane. It's a membrane that connects the rigid plates, terga and sternum in cockroach, allowing for the movement. If you have continuity of these plates without any junction, they are like joints on the exoskeleton. They are like joints on the exoskeleton, the arthrodial membrane, which gives flexibility to the cockroach body without losing its toughness. Nuclear envelope is a derivative of smooth endoplasmic reticulum, rough endoplasmic membrane of Golgi complex, microtubules, rough endoplasmic reticulum. Rough endoplasmic reticulum. We see that nuclear membrane is also in continuity with the rough endoplasmic reticulum. Does that mean there is ribosomes on the nuclear envelope also? Can there be ribosomes on the nuclear envelope as well? Can there be? There can be ribosomes attached to the nuclear membrane also, but generally it is in the rough endoplasmic reticulum. Cytochromes are found in cytochromes. What are cytochromes? Do you recall what are cytochromes? Cytochrome BCA. They are proteins which are associated with electron transport in. Is it only in mitochondria? Even in chloroplast, even in the thylakoids, you find the cytochromes as electron carriers. So in the given option, cytochromes are found in matrix of mitochondria. Is it matrix of mitochondria? Where are cytochromes attached? Electron transport chain is happening in the inner mitochondrial membrane. So cytochromes being a part of the electron carriers in the electron transport chain, where should they be attached? What should it be a part of? What should cytochromes be a part of? Cytochromes should be a part of the inner mitochondrial membrane. Inner mitochondrial membrane has what? Inner mitochondrial membrane is not given in these options. So what should we consider? Cristae. Why? Cristae are what? Cristae are the foldings of the inner mitochondrial membrane. Okay. Lysosomes are associated with what? Do you remember the sad story of lysosomes? Lysosomes are associated with digestion, intracellular digestion, clearing of debris clearance, cell digestion also. Cell digestion, programmed cell digestion is called what? Programmed cell death is referred to as, what is the term for programmed cell death, which is mediated by lysosomes. Lysosomes just rupture inside the cell, releasing all the lytic enzymes within the cell, which will break down the cell from inside and killing the cell. It's called programmed cell death. When the cell has become useless, or when the cell has become unproductive or destructive, the cell will be destroyed by the lysosome itself. When the master becomes anti-national, master has to be killed in the interest of the nation. Not loyalty to the nation at the cost, loyalty to the master at the cost of nation. No. Loyalty to the nation, kill the master if the master has become anti-national. That is your lysosome. What do you call it? Programmed cell death. Huh? Apoptosis. Hello? Hello? Alright, apoptosis. Apoptosis is associated with lysosome. Questions have come on apoptosis. What organelle is associated with it is lysosome. Which one of the following statements is incorrect? A competitive inhibitor reacts reversibly with the enzyme to form an enzyme inhibitor complex. Is it correct? Where does the competitive inhibitor bind? Where does the competitive inhibitor bind on the enzyme? It competes with the substrate for dash. Active site of the enzyme. Binding of the inhibitor to the active site of the enzyme, competitive inhibitor to the active site of the enzyme, is reversible or irreversible? Reversible. When does the inhibitor detach from the active site? You increase the substrate concentration, the inhibitor will detach from it. So statement 1 is correct. Competitive inhibitor reacts reversibly with the enzyme to form an enzyme inhibitor complex. In competitive inhibition, the inhibitor molecule is not chemically changed by the enzyme. Is that correct? Is that correct? Does the substrate binds to the enzyme, substrate is modified into product. Will the enzyme do the same with the inhibitor also? Will it change the inhibitor into some product? Inhibitor is also binding to the active site of the substrate, enzyme. Will the enzyme act on the inhibitor and change it? No. The process of inhibition brought about by the competitive inhibitor is it binds to the active site, does not get affected by the enzyme, and thereby the inhibitor will lock this enzyme, making it unavailable for converting substrate into product, reducing the efficiency of the enzymatic reaction. So statement 2 is a correct statement. In competitive inhibition, the inhibitor molecule is not chemically changed by the enzyme. Statement 3, competitive inhibitor does not affect the rate of breakdown of the enzyme substrate complex. Competitive inhibitor does not affect the rate of breakdown of the enzyme substrate complex. This is a good statement. Does it alter the rate of product formation in the entire system? In the reactor, let us say there is a reactor and you have enzymes in the reactor, bioreactor, you have substrates, competitive inhibitor is also there. Does the competitive inhibitor decrease the rate of product formation in the reactor? It does. What is the statement? Does it affect the rate of breakdown of the enzyme substrate complex? Now, this is speaking about individual enzymes. Individual enzymes. Individual enzyme, will the inhibitor binding to some other enzyme affect the rate of enzyme substrate complex changing into product? This enzyme which has got substrate will convert the substrate into product at the same duration, within the same duration that it used to do earlier also when the inhibitor was not present. So it does not alter the rate of breakdown of the enzyme substrate complex. Rate of enzymatic conversion of the substrate into the product is not altered by the competitive inhibitor. But the rate of conversion of the substrate into product or the rate of product production is altered in the entire reactor. It does not change the reaction where the enzyme is reacting with the substrate, but it will change the dynamics of the entire reactor when there are thousands of enzyme complexes. Fourth one, the presence of the competitive inhibitor decreases the Km of the enzyme for the substrate. What is Km? What is Km? Km is the maximum rate of reaction. Km is the maximum rate of reaction. Vmax is the Km is not max. Km is the substrate concentration at which the enzyme will achieve its 50% optimum conversion. That is the Km. Correct? That is the Km. With competitive inhibitor in the reactor, will the Km decrease? Will the enzymatic reactor have lesser substrate achieving 50% of the maximum rate of reaction, or will it increase? It increases because inhibitor is trying to block some of the enzymes. Inhibitor is trying to block some of the enzymes, thereby reducing the ability of the enzyme to bind with the substrate. And to achieve the 50% maximum rate of reaction, 50% rate of optimum reaction rate, you will need more substrate which can dislodge the inhibitor and make the enzyme more efficient. So correct answer is option number 4. But that is an incorrect statement. The presence of the competitive inhibitor decreases the Km or the substrate concentration at which the rate of reaction attains V50, 50% efficiency. Select the correct option. Match the matching question. Synapsis aligns homologous chromosomes. Synthesis of RNA and protein Action of enzyme recombinase. Centromeres do not separate but chromatids move towards opposite poles. Which one do you want to fix? Synapses. Synapses aligns homologous chromosomes. Synapses occurs when? Bivalent is formed in leptotene. But 2, 3, 4, 5. No, 1, 2, 3, 4. Okay. Is there something wrong in the numbering? No? Okay, synapses aligns the homologous chromosomes. In which phase? Zygotene. Leptotene is bivalent, zygotene is synapses, pachytene is recombination, diplotene is chiasma formation, diakinesis is separation. So both zygotene is synapses, recombinase is pachytene, centromeres do not separate but chromatids move towards opposite poles. That is anaphase 1. That means what is moving here? It is not the sister chromatids are being separated, it is the homologous chromosomes are being separated. In anaphase of mitosis, what separates? If anaphase of mitosis was the option, what would have been the statement? Change the statement. Statement D. Change it to make the anaphase of mitosis the right choice. Right match. No, change the statement, full statements. Centromere disintegrates and the sister chromatids move to the opposite poles. Wouldn't that be the right statement? To make anaphase of mitosis the matching option? Alright, so it is synapses during zygotene, synthesis of RNA and protein during G2 phase, action of enzyme recombinase in pachytene, and centromeres do not separate but chromatid move towards the opposite poles in anaphase 1. A somatic cell that has just completed the S phase of its cell cycle. Are you noticing cell cycle and cell division? Almost 2 to 3 questions in every paper. All standard. The concepts are same. Every paper you should get it. A somatic cell that has just completed the S phase of its cell cycle as compared to a gamete of the same species has twice the number of chromosomes and twice the amount of DNA. Okay, see, look at the question again. Somatic cell that has just completed the S phase of its cell cycle. So somatic cell, we are looking at it in the S phase as compared to a gamete of the same species. We are not looking at a cell in the S phase. We are not looking at a cell in meiosis. On one side, we are looking at a cell in mitosis because it is somatic cell and in the S phase, which has completed S phase. On the other side, we are looking at a gamete. Now, don't look at the options. Give numbers. Let us say a somatic cell has 10 chromosomes and 10 units of DNA. Somatic cell has 10 chromosomes and 10 units of DNA. What will be the number of chromosomes and the unit of DNA in a gamete? Gamete of the same species will have how many chromosomes? 5 chromosomes, half the number of chromosomes. And how many units of DNA? 5 units of DNA. Alright? This somatic cell goes into mitosis and has completed S phase. What has happened? And it has completed S phase just now. DNA content doubles. So the DNA content will be 20 units. Number of chromosomes will be, technically it would be 0. In a somatic cell also chromosomes will be 0. You will not see any chromosomes. But for the sake of discussion, number of chromosomes will be 20. Chromosomes also doubles and DNA content also doubles when the somatic cell completes S phase. So compared to gamete, How many times the number of chromosomes are there? 4 times. 4 times the number of chromosomes, and the DNA content 4 times. Look at the options now. Twice the number of chromosomes and twice the amount of DNA. Same number of chromosomes but twice the amount of DNA. Twice the number of chromosomes and 4 times the amount of DNA. 4 times the number of chromosomes and twice the amount of DNA. Twice the number of chromosomes and 4 times the amount of DNA. It is not 4 times the number of chromosomes. Alright? But, no, it should be 4 times the number of chromosomes, no? See, the options are wrong. Somatic cell, 2 chromosomes. 2 chromosomes. Gamete will also have 2 chromosomes. Chromosomes. 2 chromosomes. S phase, four chromosomes. Gamete, 2 chromosomes. Correct. Twice the number of chromosomes and 4 times the number of 4 times the DNA content. 4 times the DNA content. 2, 4, 6, 8 DNA content. Each line is 1 unit of DNA if you assume. The somatic cell had 4 units of DNA. Gamete had 2 units of DNA. After the S phase, doubling has happened. So it has 8 units of DNA. But the doubling has created sister chromatids for each chromosome. It is not separate chromosomes. So the number of chromosomes are still 4, with each of the chromosomes having double the content of DNA. It's a good question. Twice, did you understand? Did you all understand? No? No? After DNA duplication, see, do you still remember the concept of chromosome, chromatid, sister chromatid, homologous chromosomes? A unit of condensed chromatin is chromosome. 1 unit of condensed chromatin is chromosome. How many units of condensed chromatin are there here? Each one is a separate chromatin thread. There are 2 units of condensed chromatin. So how many chromosomes are there? 2 chromosomes. Here, just look at this. There are 2 chromosomes. Each of the chromosome duplicates. Forming 1 chromatin thread becomes 2 chromatin threads. But both the chromatin threads are remaining together. Content of DNA became double. But how many chromosomes are there? It is still 1 chromosome only. Here, each of these chromosomes doubled the DNA content. But both the chromatin threads are remaining together, forming twice the amount of DNA, but 1 chromosome only this is. So there are 4 chromosomes here. After the S phase also, you have 4 chromosomes, but each of the chromosome has a duplicate copy of itself associated with it. Is that clear? Each chromosome has a duplicate copy associated with it. Now, each of these copies in the chromosome are referred to as chromatids. You don't call them chromosomes anymore. When you separate them and each of these threads become independent, you call them chromosomes. Inherent problem with this question is, we should not be talking about chromosome except during metaphase, except during the mitotic phase. End of prophase you see chromosomes, metaphase you see chromosomes, anaphase you see chromosomes. Beginning of telophase you can still see chromosomes, but it will chromosomes will decondense and become chromatin mass. But for the sake of the question, we have to consider this. Alright. So twice the number of chromosomes and 4 times the amount of DNA. Twice the number of chromosomes at the end of. Yeah. Correct, no? Twice the number of chromosomes and 4 times the amount of DNA. Which of the following statements is not correct? Digestion is anyways not there, no? The syllabus. Let's see this once. Or we'll skip it? Skip it. Huh? Skip it. We'll skip the discussion on questions which are not there, which are not a part of the syllabus. So all the, just for the sake of knowledge, time is over. Now it is only for the purpose of exams for the next 3 months. Gastric juice of infants contain nothing. When you hold your breath, which of the following gas changes in blood would first lead to the urge to breathe? Which gas is a trigger for initiating breathing? Where? Where do you get the trigger for breathing? What do you call that? And
Pneumotaxic center.
Pneumotaxic center, where do you find it? In the medulla oblongata. Medulla has the vital centers, pneumotaxic center and one more.
Rhythmic.
Huh?
Rhythmic center.
What? Rhythm center, what do you call that? Vasomotor center. Vasomotor center and the pneumotaxic center are present in the medulla oblongata. What stimulates medulla? If you are holding breath, 2 things are happening. Oxygen level is going down, carbon dioxide level is increasing. Which of this is a stronger trigger for initiating breathing? What will make you just leave your breath and inhale? That is brought about by which gas? Falling oxygen concentration, rising carbon dioxide concentration. Falling carbon dioxide concentration. Option 4 is giving you both rising carbon dioxide and falling oxygen. Safe players will choose that. Both are happening anyways, so we will choose the option 4. No, but which is the trigger? Carbon dioxide is the trigger. Carbon dioxide is the trigger, not oxygen. How does the pneumotaxic center pick up carbon dioxide level? Because carbon dioxide level is not directly identifiable. There are chemoreceptors in the blood which will sense the acidity of blood, which will sense the pH of blood, acidity. Carbon dioxide accumulation will lead to acidosis, acidosis of the blood, metabolic acidosis. Acidosis is sensed and then the breathing is triggered. Blood pressure in the mammalian aorta is maximum during. Blood pressure in the mammalian aorta is maximum during when systole of the left atrium, diastole of the right ventricle, systole of the left ventricle, diastole of the right atrium. This is an easy question. When does the aorta experience high pressure? When does the aorta experience the highest pressure, blood pressure? There during systole of atria or systole of ventricle or when? Systole of ventricle. Which ventricle? Left ventricle. Alright, next one. Which one of the following is correct? Plasma is equal to blood minus lymphocytes. Serum is equal to blood plus fibrinogen. Lymph is equal to plasma plus RBC plus WBC. Blood is equal to plasma plus RBC plus WBC plus platelets. Plasma is blood minus all the formed elements. What are formed elements? WBC, RBC, platelets. WBC, RBC, platelets. Lymph is you can consider serum plus nutrients. Serum plus nutrients. Serum is what? Serum is plasma minus the clotting factors. Plasma minus clotting factors is serum. Alright. Then what else is there? Okay, lymph we discussed, plasma, serum, blood. Okay. What is the right choice here? Plasma, blood is plasma plus RBC plus WBC plus platelets. Blood products was the first presentation I made during first year MBBS in physiology. I don't like keeping paper and reading from the paper, however long the presentation is. I want to just do it impromptu. So during the presentation, everybody were holding their notes for reference. I did it impromptu, 20 minutes. So that is the presentation when I made some impression. All right. Whether I lived up to that impression or it went down or it went up, that's all different story. But impression was created. Blood products. Those were the times when, say, separation of blood products for targeted treatment was not yet so prominent. Now we tell, no, a unit of blood donated saves 4 lives. You separate the blood products, blood components, you separate plasma, you separate clotting factors, you separate the RBCs, all that, serum and all that. Removal of PCT from the nephron will result in what? More diluted urine, more concentrated urine, no change in the quality and quantity of urine, no urine formation. What will happen if PCT is removed? Huh? PCT is responsible for what? PCT is responsible for what? Reabsorption of nutrients, then ions also. Nutrient and ion reabsorption happens predominantly in the PCT. Certainly there will be more urine volume. Urine volume will be much higher. But will it be more concentrated or dilute is the question. If nutrients are not absorbed, the nutrients are remaining in the PCT, in the renal tubule. Nutrients are remaining within the nephron. When it comes to the Henle's loop, what will happen to the countercurrent multiplier mechanism? It alters the ion absorption dynamics because the osmolarity balance is lost. The osmolarity balance is lost. So, reabsorption of water is also affected significantly. And when it comes to DCT, predominant reabsorption of water, nutrients, everything should have been completed in PCT and Henle's loop. But when it comes to DCT, again a higher concentration of nutrients, higher concentration of osmotic substances in the DCT will further impair the reabsorption of water from DCT. So what will be urine? Will it be more concentrated, more dilute? It will be more dilute, high in volume and more in dilution. Because if you look at it logically, PCT did not absorb nutrients, so more osmotic substances are present in the tubule. Rest of the things do their job properly. So more water absorption happens in the Henle's loop and the DCT. Urine should get more concentrated. It doesn't happen because absorption in the Henle's loop and the DCT is dependent on the osmotic gradient, osmotic pressures. That gradient is disturbed and the absorption is also hampered. So more dilute urine will be produced. Sliding filament theory can be best explained as when myofilaments slide past each other, actin filaments shorten while myosin filaments do not shorten. Does any of the filaments shorten? No filament shortens. So if anything talks about filaments shortening, that is a wrong option. Check for it. Actin and myosin filaments shorten and slide past each other. Statement 2. Neither of them shorten. So statement 2 is also wrong. Actin and myosin filaments do not shorten but slide past each other. That is the right explanation of the sliding filament theory. When myofilaments slide past each other, myosin filaments shorten while actin filaments do not shorten. Again, baseline you need to know in muscle contraction, there is no shortening of either of the filaments, actin or myosin, or the thin filament or thick filament. Alright. So sliding filament theory talks about calcium contributing to the attachment of the head of the myosin to the thin filament, and then ATPase activity of the thick filament breaks down the ATP, releasing energy which brings about flexion of the neck of the myosin head, pulling the thin filaments inside. Alright. My sister is working on sliding filament theory. Her research is on sliding filament theory. She is working on something which has the potential to disprove or find fault with the sliding filament theory. Alright. So if you find something wrong with, she is not the primary researcher there, she is working with a researcher professor there whose work is to disprove sliding filament theory. So if that gets validated and he is able to prove that, he would probably be getting a Nobel Prize. Professor Dr. Josh Baker. She will also be a part of the team which worked on that. Alright. Whether it will happen or not, that we don't know. I don't discuss her research much with her, but she is working on sliding filament theory. To dispute it, not to understand it. A lot of experimentation to say sliding filament theory is wrong. Alright. Glenoid cavity articulates. Where do you find glenoid cavity? Where do you find glenoid cavity? It is the cavity in which the head of the humerus articulates with the scapula. It is between humerus and scapula. Shoulder joint or the shoulder joint is the most versatile joint of the human body. Most versatile joint. Now with the versatility comes regulation, micro-regulation. With more regulation comes more innervation, nerve supply, more ligaments, more muscles attached to it, smaller and bigger muscles, all of that. Damage to shoulder leads to more complex repair. Sportsmen damage their, cricketers are more likely to damage their shoulders and all. They go through surgeries, 3 to 6 months of recovery. Which of the following regions of the brain is incorrectly paired with this function? Medulla oblongata, homeostatic control. Is it correct? Does medulla contribute to homeostasis? It does by regulating respiration and heart rate. Cerebellum, language comprehension. Is it right? Language is associated with what? Language is associated with what? Cerebrum. There are functional areas in the cerebrum. There are two types of areas in the cerebrum. Functional areas and what is the other type of area? Two types of areas in the cerebrum. Functional areas and association areas. Association areas are like, it's a place of collaborative analysis. It's like the research center. Functional areas are associated with individual functions. There are two areas which regulate speech. Language is regulated by two areas, Broca's area and Wernicke's area. Broca's area is associated with articulation, innervating your vocal cords, mouth muscles and all that, controls all that, so that you can produce sound. Sound production is controlled by Broca's area. Sound meaning is controlled by Wernicke's area. So speech has two components, motor speech and sensory speech. Motor speech is the ability to create sounds. Sensory speech is the ability to make sense. Alright. Absence of speech is referred to as aphasia. Motor aphasia and sensory aphasia. People who are not able to articulate, sound mechanism is defective, it is called motor aphasia. Sensory aphasia you see every day, night, 9 p.m. Most of the news channels, political debates. People speak continuously, but there is no sense. Okay? That is sensory aphasia. What you speak, they will not be even listening to it, so they don't understand or comprehend the other person's speech, but they continuously produce speech. Sensory aphasia. Very difficult to handle people with sensory aphasia. They will not stop. You tell a sentence, they will find 10 points in one sentence to create a half an hour speech. Some nonsense. Horrible. So you should learn to identify people with sensory aphasia, and your best approach to dealing with them is not to engage in conversation. Alright? You will not win. Debating with a fool has got only one option. Whether you win or lose, you are more likely to become a fool. A gymnast is able to balance his body upside down even in total darkness because of cochlea, vestibular apparatus, tectorial membrane, organ of Corti. Come on, yeah. What helps you identify your position? Position sense is because of what? Again, special sense organs. You would say out of syllabus now. Let us skip it. Vestibular apparatus, semicircular canals. Organ of Corti is responsible for converting sound waves into, or the mechanical waves into electrical impulses. Okay. Good thing is elimination of a lot of botany topics. Mineral nutrition, transport related things. One disappointment I still have is they still have the 5 kingdom classification, biological classification. All those 3 chapters should have been eliminated. Correct, no? Biological, all the 5 kingdoms should have been out of syllabus. That would have made medical aspirants prepare for their first year of medical course much more efficiently. You should have only physiology, reproductive biology, genetics, evolution, molecular biology, cell biology, ecology also, because that also has significance for your medical course. That's all. And application, biotechnology and practical application, biotechnology and applied biology. That's all. No? That should be the syllabus for NEET. I think it is moving towards that. It is moving towards that. Very shortly we should have only this. Global medical entrance exams have only this. Only our exam has everything. They are anyway studying all these things. Let them, right? That is the approach. A chemical signal that has both endocrine and neural roles is. Chemical signal that has both endocrine and neural roles. Melatonin, is it endocrine? Melatonin, is it a hormone? Endocrine roles means it's a hormone. Neural role means it is a neurotransmitter. You have to identify a chemical which is both a hormone and a neurotransmitter. Alright? Melatonin, is it a hormone? It is associated with sleep-wake cycles. Calcitonin, is it a hormone? Calcitonin is a hormone, regulates what? Tell me one function of it. Melatonin is not a neurotransmitter. Calcitonin is also not a neurotransmitter. The question is easy. You have it is epinephrine, which is adrenaline, which is both a hormone as well as a neurotransmitter. Epinephrine, norepinephrine are neurotransmitters also. Alright. Calcitonin, what does it do? Tell me one function of calcitonin. Primary function of calcitonin. What does it do? Increases blood calcium or decreases blood calcium? Decreases blood calcium. Calcitonin is bone making hormone. So remove calcium from blood and push it into bone. Counter hormone for calcitonin is parathyroid hormone or parathormone. Increases blood calcium. Which of these two hormones when hypersecreted cause osteoporosis? Which of these two hormones, calcitonin and parathormone, when hypersecreted cause osteoporosis? Parathyroid hormone. A 50-year-old man comes with fracture of let's say some bone and gives a history of repeated fractures. An endocrine profile done of this person is more likely to show hyperactivity of which gland? A person, a 50-year-old male showing history of repeated fractures due to not so significant injuries. Which gland hyperactivity can be the likely cause of this? Parathyroid gland. See, same thing. You can keep on asking the same thing in different ways. You should be able to correlate. Think. Logically think. Understand the question, think. Cortisol counters which metabolic hormone? Cortisol counters the action of which metabolic hormone? Insulin. Cortisol counters the action of insulin. Alright. Is the role of cortisol similar to that of glucagon? Partially yes, because it increases blood glucose level, but completely not right, because cortisol can increase blood glucose levels much higher than the range of glucagon. And cortisol is not inhibited by the presence of insulin. Insulin and glucagon are counter hormones. When insulin is present, glucagon is not working. When glucagon is there, insulin is suppressed. So cortisol, that one. Which one is the stress hormone of the four given here? Stress hormone, hormone that is released during stress. Cortisol. Which is the hormone released during flight or fright response? Adrenaline or epinephrine. Which of the following does not favor the formation of large quantities of dilute urine? Which does not contribute to formation of large quantities of dilute urine? Alcohol, does it contribute? Yes. Alcohol increases urination. It causes diuresis. Alright? Oh. A session of 2 hours, 3 hours, making some of you want to go to the washroom for a urine break every hour. Water. Caffeine. What does caffeine do? Caffeine also increases urine output. Renin. What does renin do? Does renin increase urine output or decrease urine output? Why? Renin is a part of renin-angiotensin-aldosterone system. Renin contributes to water retention and sodium reabsorption, reducing the volume of urine produced. So, more renin will lead to more concentrated urine, not dilute urine. ANF, atrial natriuretic factor. It increases sodium excretion, thereby increasing the urine output. So what is the right choice here? Renin, which does not favor formation of large quantities of dilute urine. Capacitation refers to changes in the sperm before fertilization, ovum before fertilization, ovum after fertilization, sperm after fertilization. Very simple question, basic question. Capacitation is what? Sperm before fertilization. Capacitation, I'll change the question. Capacitation happens when the sperm is in the vas deferens, is in the urethra, is in the vaginal tract, is in the fallopian tube. When sperm is in the vaginal tract. So capacitation is initiated when semen is exposed to the vaginal fluid. When the semen liquefies and sperms are activated, then capacitation happens. With what ion is required for capacitation of sperms? What ion is required for capacitation of sperms? Calcium. Calcium is required for capacitation. What is the source of energy that is loaded into semen for the survival of sperms during its journey from the vagina to the ovum? Fructose. Alright? So you should remember fructose and calcium. Fructose is the source of energy. Calcium is required for capacitation. Which of these is not an important component of the initiation of parturition in humans? Increase in estrogen and progesterone ratio. Is it important? More estrogen, less of progesterone. Increase in estrogen progesterone ratio. So when relatively progesterone becomes lesser and estrogen becomes more, it triggers placenta to release more prostaglandins. It triggers the corpus luteum to produce relaxin. These are all triggers. It activates the hypothalamus to produce and trigger the posterior pituitary release oxytocin. Synthesis of prostaglandins. Is it important for initiation of parturition? Yes. Who synthesizes prostaglandins? Who synthesizes prostaglandin? Huh?
Pituitary.
Prostaglandin is produced by pituitary gland. Very good. We have discussed this. Yesterday we discussed. Yesterday or day before. 2-3 days ago we discussed this. Three hormones are three chemicals are responsible for increasing contractions of the uterus. What are the three chemicals? Two chemicals let us consider. Oxytocin and prostaglandin. One is responsible for relaxation of the pelvis wall muscles and ligaments. What is that? Relaxin. Oxytocin is produced by pituitary. Oxytocin is produced by hypothalamus, released from posterior pituitary. Prostaglandins are produced by placenta only. Placenta produces prostaglandins. Is prolactin required for parturition? Prolactin is not required for parturition. Prolactin is required after parturition. Prolactin is released during parturition only. Prolactin will get stimulated to keep the mother's mammary glands ready for feeding the baby post delivery, but very small quantities. It is ready, it is on standby, but it is not required for parturition. Which of the following viruses is not transferred through semen of an infected male? Hepatitis B virus, HIV, chikungunya, Ebola. Other than chikungunya, which is transmitted by who? Aedes aegypti mosquito. Aedes is responsible for both dengue and chikungunya. Alright, chikungunya affects the muscles, causes terrible pain in the muscles and joints. All the other three infections happen via coming in contact with the infected fluid. Which of the following cells during gametogenesis is normally diploid? Primary polar body, is it diploid? Primary polar body is produced when? At the end of meiosis 1. At the end of meiosis 1, primary polar body is produced. So is the primary polar body diploid? Meiosis 1 gives rise to haploid cells. So primary polar body is haploid. One is the secondary oocyte, and the other one is primary polar body. Both are haploid. Spermatid, is it diploid? Spermatid is the cell that is produced at the end of meiosis 2. Tell me the sequence of cells starting from spermatogonia and their ploiding. Spermatogonium, diploid, undergoes growth or metabolic modifications. Growth phase is not much in spermatogenesis. Then after spermatogonia it is primary spermatocyte. Meiosis 1, primary spermatocyte is diploid again. Meiosis 1 gives rise to secondary spermatocytes, which are haploid. Secondary spermatocytes complete meiosis 2, produces spermatids, haploid. Spermatids will undergo spermatoeleosis or spermiogenesis to produce sperms or spermatozoa. Spermatozoa and spermatid are not the same. Sperms are spermatozoa. Spermatid is the precursor. Both of them are haploid though. Alright. Hysterectomy is the surgical removal of uterus, prostate gland, vas deferens, mammary glands. Tell me the names for each of these. Surgical removal of each of these, what is the name? Uterus is hysterectomy, done. Prostate gland is?
Prostatectomy.
That's all, prostatectomy. Vas deferens is? Vasectomy. Ectomy. Mammary glands is? Mammary glands is? You can say that. Mammectomy. Alright. That's all. You put ectomy. Removing appendix. Removing appendix. Not appendix, appendixtomy. You have to make it sound also good, no? You should sound like one word. Removal of appendix is appendicectomy. Alright. Removal of stomach. Removal of stomach is done. It is called bariatric surgery. Why do you remove stomach? Okay, I'll give you an option to remove stomach of somebody. Who is that somebody whose stomach you will remove? It cannot be based on your personal likes and dislikes, friendship and enmity. It has to be based on sound medical judgment. Whose stomach you should want to remove? You would want to remove.
Stomach cancer.
Okay, stomach cancer. Excellent, good. Okay, no cancer. Then, who else? Very good. Stomach cancer. They have stomach cancer. You have to remove stomach. Remove stomach. Different molecular forms of a gene, heterozygotes. It's a straightforward question out of your definition of what alleles is. Alleles are different molecular forms of a gene. Different phenotypes are caused because of different alleles of the same gene. Yes or no? Can we say that? Variation in phenotype is caused because of different alleles of the same gene, or different phenotypes are caused because of different alleles of the same gene. Should we consider phenotype to be equivalent to the character, or the variations also to be considered as phenotype? Tall and dwarf in pea plant, should we consider them as two different phenotypes? As a character, we look at it as height. So gene is for height, alleles are for different phenotypes. Character is represented by gene. Phenotypes, variations are represented by alleles. So why not alleles are different phenotype be the right choice?
Because phenotype is sometimes because of environment.
Phenotype is caused because of the differences in the alleles. Alleles are not phenotypes. The question is, what are alleles? Okay. True breeding homozygotes are what? True breeding homozygotes are homozygous alleles. Homozygous alleles crossed with the different homozygous individuals only. So dominant homozygous recessive crossed with homozygous recessive is a true homozygotes. So that is not a definition of allele. Anyways, let's move forward. This is heterozygotes are those which have both the varying alleles. A man with blood group A marries a woman with blood group B. What are all the possible blood groups of their offspring? Tell me. All four are possible. See, we don't know whether the man and woman, either of them or both of them, are homozygous or heterozygous. But because it is not mentioned, we have to look at all possibilities. So the man is IAI, if at all this is the group. The woman is IBI, if this is the group. If either of them is homozygous, then what is not possible? If either of them is homozygous, O blood group is not possible. And because nothing has been specified, we have to assume the maximum possibility. The maximum possibility is when both of them are heterozygous, which gives us all four as the probable blood groups in the offspring. Gene regulation governing lactose operon of E. coli that involves the LAC-I gene product is. LAC-I is what? What is LAC-I? We have seen LAC-ZYA. What is LAC-I? The gene which produces the repressor protein. It is the inhibitor gene which produces the repressor protein. Now look at the options. Gene regulation governing lactose operon of E. coli that involves the LAC-I gene product. LAC-I gene product is repressor protein. What is the gene regulation is the question. Is it a positive gene regulation, positive and inducible, because it can be induced by lactose. Negative and inducible because repressor protein prevents transcription. Negative and repressible because repressor protein prevents transcription. Same options, huh? 2 and 3.
Inducible and
Inducible and repressible, correct. Feedback inhibition because excess of beta galactosidase can switch off transcription. Lac operon, lactose metabolism is regulated by what kind of operon? Is it an inducible operon or a repressible operon? It is inducible operon. That means what? By default it is repressed, it has to be induced. Repressible operon is, by default it is active, it has to be repressed. Alright. So, what is your by default status? To study or to sleep? By default, we want to rest, we don't want to put in the hard work. Mind always wants the comfort zone. You have to be induced into putting in effort which will push you beyond the comfort zone. Like addiction, whatever you consistently hold will become your baseline. Comfort zone expansion also happens like that. You constantly push your comfort zone. That expanded zone, which earlier was a discomfort zone, the periphery of that was discomfort zone, you stay there for a period of time, that becomes your comfort zone. So on a daily basis, you should not be in a state of comfort. Every day you should push yourself beyond something, beyond what you are actually comfortable with. So during training, I always tell people, see, studies, doing well in exams, all of that are a consequence of who you are. Who you are is a consequence of what you do on a day-to-day basis, which is mostly what you do. Don't eat unless you cannot stay hungry no more. Don't sleep unless you can stay awake no more. Perfect. Whenever you feel like this is the limit, do a little bit more. You are exercising, you are doing Surya Namaskara, you do 12 and you want to push it. You are somewhere coming up to 47, 48, push a little more, do 50. When you feel this is the limit, just one more, just 2 more. That's all. In everything. So you should push yourself to the point where your body starts telling, no, not anymore. Then hold for a while. That's all. That is continuous training of your mind to expand your comfort zone. Every day, your 24/7 can be your training in mindset, training in your tendencies. Alright. And all of that is what will help you perform. Focus should be yourself. Continuous focus is yourself. Changing focus is whatever is the priority at any particular point of time. Now the priority for the next 3 months is studies. After that, for about 2 to 3 months, till you get results and all your counseling is done, is spending your parents' money, whatever you can, in whatever form of entertainment and enjoyment you want. Then keep changing. So it is negative and inducible because repressor protein prevents transcription. In sea urchin DNA, which is double-stranded, note down the key data points. Double-stranded DNA, 17% of the bases were shown to be cytosine. The percentage of the other 3 bases expected to be present in the DNA are, even before you calculate the percentage of other nucleotides or other bases, which one do you want to conclude just like that? Guanine. How much should guanine be?
17.
17. So check for the options where guanine is not 17. 1 and 4. Eliminate it. Whatever you eliminate in your paper, you can write on your paper, no? You can mark on your paper. The options which you have definitely eliminated, strike it off. Strike it off so that it does not distract you while you are analyzing the other options. Alright? Otherwise, what happens? Your option 3 is wrong at the third point, let us assume. But at the third point, option 4 is right. And these two are next to each other, very close to each other. If you are not striking off option 4, which is the wrong option, the mind can read 1, 2 from option 3, 3 from option 4 and 4 from option 3 and tell you option 3 is right. Mind has a unique ability of filling the blanks. You use information and try to create the right create an impression. So you are sure that something is not an option, strike it off. It will save time. Saving even 10 seconds of time in that in overcoming that distraction or avoiding the distraction is advantage. 10 seconds is advantage. This is all part of your strategy. Okay? So 17 plus 17 is 34. Rest of it is 16, 6. The other two should be 33. That's all. 33, 33. And even without calculating, you can still conclude between 2 and 3. How do you conclude without even calculating? Adenine and thymine should be equal. That's all. Forget about calculation also. Adenine and thymine are equal only in one option, in two options, but the first option is wrong. Third option is right. What is the rule that determines this? Name the rule. Chargaff's rule. Chargaff gave the rule. What is the rule of? It is the Chargaff's rule of base equivalence. Chargaff's rule of base equivalence. That is the rule. Which of the following had the smallest brain capacity? Had the smallest brain capacity, our ancestors. It was there, this question only. Yeah. You mark it right? Just buying the ticket after the train has left the station. If we had discussed this yesterday, you would have got this correct today. See, you should know the highest brain capacity was of Neanderthals. Neanderthals were supposed to have even bigger brain capacity than us now, Homo sapiens. So second highest is of Homo sapiens. The earliest, other than that, every ancestor of ours, from the earlier one to the later one, has gradually increasing brain capacities. So of the four, you arrange them in the order of their antiquity. Whoever is the most ancient to the most recent. Who is the most ancient of the four? Homo habilis. Then Homo habilis is the what man? Tool maker. Homo habilis is the tool maker man. Started using stone tools and all the small tools. Then Homo erectus. Then Homo neanderthalis. Then what we are now, Homo sapiens. Homo erectus about 600 to 800 cc, cubic centimeter. Homo neanderthalis around 900 to 1100. Homo erectus. Habilis 600 to 900, 1000 to 1100 is erectus. Around 1300 to 1400 is Homo sapiens. 1400 to 1500 is Homo neanderthalis. Does that mean we have become less intelligent than our immediate ancestor, Homo neanderthalis? We don't know, probably. Population will not exist in Hardy-Weinberg equilibrium if. Tell me the conditions for Hardy-Weinberg equilibrium to exist. Then we will look at the options where it says it does not remain in equilibrium.
No gene mutation. No
Those who are not in the room now should be noted down. Madam, please make a note of the students' names. Those who are not in the hall now. With whose permission have they stayed out? Okay. Others? There are about 5-6 of them not in the hall. two things I don't like. Number one, misusing my lenience. Number two, taking me for granted. Those two will create problems. Alright? Don't misuse lenience. Don't take me for granted. The more lenient I am, the more dangerous I am also. Okay? Not that I'll come and beat you all up. But no. That's all. Apart from those two, you don't take me for granted and do things, hey, it's okay, chalega kind of attitude. That is why any deviation from the process irritates me. That is taking things for granted. I say it has to happen this way, it has to happen that way. No deviation. If you feel something else would be better, you have to discuss, convince me, and get the change approved. Until then, no deviation. Right. So when I am here, how does my school run? Any deviation, people know, they will have to face some crazy criticism. So no deviation. If somebody feels something else is better, go create it. No option to not follow what I create in the system that I create. Okay. Whoever is the roommate of those people who are absent now, those people have to be informed, they have to talk to me tomorrow. Alright? Note them. What are the conditions in which Hardy-Weinberg equilibrium exists? Population should be large population, then no migration, then random meeting, then anything else?
Mutations.
No mutations. These are the four conditions. If any of these four are disturbed, then Hardy-Weinberg equilibrium will be disturbed. Let us look at it. Individuals mate selectively. Equilibrium or disruption? Disruption. No mutation, no migration, population is large. All of those are favorable for equilibrium to exist. Disease with the type of vaccine. Tuberculosis. What is the bacteria? What is the pathogen for tuberculosis? Tubercle bacillus. Bacillus tuberculosis. The vaccine is? It is a whole vaccine of Mycobacterium tuberculosis, which is killed. It is a killed whole vaccine. What is the vaccine? What is the name of the vaccine for against tuberculosis?
BCG.
BCG. Bacillus calmette-guerin. Named after the scientists who created this vaccine. What is the mode of given the vaccine? It is intradermal. The vaccine, the bacteria is put under the skin, in the dermis of the skin. It will cause a blister, bleb. All of you have this scar, no? All of you have the scar, which is BCG vaccine scar. Some people have bigger scars. Big, almost like 50 paisa coin scar. What is that scar? That will be on the other side, not on this side. What scar is that? Not BCG. BCG will cause only the small scar. The bigger 50 paisa coin scar. Have you seen that?
Yes.
Big scar. What is that? If you look at that scar, you can tell when that person was born. At least you can get the cutoff. Those who are born before 1982 have that scar. Those who are born after 1982 don't have that scar. So at least a basic age cutoff you can make by the scar. What is that scar? Come on, nobody knows it. Smallpox. It is smallpox vaccination scar. Earlier, smallpox vaccine used to be given, but smallpox was eradicated, and after that, vaccination against smallpox is not being given. Smallpox vaccine used to cause that big scar. Maybe your parents might have big scar. Grandparents will definitely have. Okay. So tuberculosis killed bacterium. Tuberculosis killed bacteria. A3. Whooping cough. Whooping cough and diphtheria both are inactivated toxins. Inactivated toxins. Polio is harmless virus. Whooping cough and diphtheria. Whooping cough is caused by what? Whooping cough is caused by? Whooping cough is also has a name. What is the name? Pertussis. Bordetella pertussis. That is the pathogen. Diphtheria is caused by? Clostridium diphtheriae. Alright, inactivated toxins. Toxoids are given. Polio. There are two types of polio vaccines. One oral polio vaccine and the other one is injectable polio vaccine. Oral polio vaccine is called Salk vaccine, SALK, Salk vaccine. The injectable polio vaccine is called Sabin, SEBIN, Sabin vaccine. May not be asked to that detail, but if you know in the eradication of polio, we have already eradicated that from our country. Polio cases are no longer reported in the country. A few cases are being reported every year, 1 or 2 cases from Pakistan. So it is declaring polio as being eradicated. Okay? That's all. HIV that causes AIDS first starts destroying what? Helper T cells. What is the cell differentiation marker that you find on helper T cells? Cell differentiation marker. So by that, helper T cells are also referred to as CD, CD4, 8, CD8 T cells. CD4 is killer. Okay. CD4 is helper. CD4 is helper, CD8 is killer. CD4 plus helper cells. Active form of Entamoeba histolytica feeds upon. Active form of Entamoeba histolytica is a trophozoite. Feeds upon the mucosa and submucosa of the colon. Red blood cells. It eats red blood cells. Entamoeba histolytica is what? What does it cause? Amoebiasis. Initially, it will start as amoebic dysentery, and then once it gets into the bloodstream, it colonizes which organ? See, the Entamoeba histolytica will start scraping on the intestinal mucosa. It will start biting on the intestinal mucosa, chops off the intestinal mucosa, then goes onto the wall of the intestine, bites onto that, eroding the wall of the intestine. As the intestinal wall erodes and this reaches the blood vessels, small capillaries, bleeding starts. It will eat the blood also. It is doing that to eat blood only. Entamoeba histolytica. And once it gains access into bigger blood vessels and lymphatics, it gets into the lymphatics. Via circulation, it goes and reaches where? Where does it go and settle down? It is eventually trying to go and settle down in one place, which is that place where the amoebic parasite wants to settle down. Liver. Amoebiasis happens in the liver, where this amoeba forms, they form colonies. They form colonies and covers themselves with a cyst. So you will find amoebic cysts within the liver. So these cysts are space occupying lesions within the liver. They occupy space, reducing the efficiency of liver function. Liver function will get affected. And as the cyst enlarges with more and more amoeba becoming a part of that cyst, at some point of time this cyst will rupture. Amoebic cyst will rupture and these parasites can enter the bloodstream. When they enter the bloodstream, they can move to different parts of the body and settle down in various other organs also. Let's say it reaches into the brain circulation. Can it go and enter the brain directly? It can't enter the brain because there is a blood-brain barrier. There is a blood-brain barrier. But what is the tendency of this fellow? What is the nature of this fellow? Eating on the wall wherever it is attaching to. If it goes and gets stuck at the blood-brain barrier, it wants blood to eat. That's all. It will start eating and it will start scraping the wall of the blood vessels. At some point of time, it will break the blood-brain barrier and it can enter the blood circulation. It can enter the brain circulation. Once it gets into the brain circulation, it can settle down wherever it can get the opportunity to settle down. So there can be amoebic cysts formed in the brain also. See, you can say any day, anyways, everybody has to die someday. So what if they die by this way? Alright, but it's all very causes it causes significant disease. And how does this come in? How does this enter? Entamoeba histolytica infection happens by contaminated food and water. You just have to ensure that you eat good food, clean and hygienic food, and drink safe water. That's all, you will be taken care of. Eating anywhere, eating whatever you find, wherever it is, you will not die if you don't eat for a day. But if these fellows get in, they will play with you for many years. And even with treatment, this amoebic Entamoeba histolytica cannot be cleared off completely. Removing parasites from their resting spots is very difficult. They can stay. Alright. High value of BOD. What is BOD? Biochemical oxygen demand indicates that water is pure, water is highly polluted, water is less polluted, consumption of organic matter in the water is higher by microbes. Water is highly polluted. Pollution by what? What pollution is indicated by BOD? Pollution of organic matter. So how can organic matter be a pollutant? What will contribute to organic matter pollution? Sewage. Sewage. Lot of fecal matter. Fecal matter is also organic matter only. Organic matter only. So feces containing sewage let out into water bodies will lead to increased organic content in the water bodies. Bacterial growth happens because there is a lot of food available for them, organic matter available for them. Biological oxygen demand increases because of increased metabolism of all the microbes present in the water body. Most animals are tree dwellers in a coniferous forest, thorn woodland, temperate deciduous forest, tropical rainforest. Tree dwellers will be more when there are a lot of trees. When the entire vegetation is covered by large number of trees, large number of organisms will be tree dwellers, arboreal, will be arboreal. So in which of these four you are more likely to find more tree dwellers? Tropical rainforest. Following graph depicts changes in the population of herbivores in a grassy field. A possible reason for these changes is, just a moment, I don't see the graph, I'll pull out the graph. Following graph depicts changes in two populations of herbivores in a grassy field. A possible reason for these changes. Which one of the following is wrong for fungi? Pretty straightforward. Are they eukaryotic? See, in these questions, negation questions, you have to be careful. Which one of the following is wrong about fungi? What is wrong about fungi is the right choice. Fungi are eukaryotic, so that is right about fungi, so not the right choice. All fungi possess a purely cellulosic cell wall, is that right? Fungal cell wall is chitinous. Bacterial cell wall is peptidoglycan. Bacterial cell wall is peptidoglycan, fungal cell wall is chitin, plant cell wall is cellulose and lignin in the secondary cell wall. So this you have to remember. Heterotrophic, yes, fungi are saprotrophic, both unicellular and multicellular, correct. Methanogens belong to Eubacteria, Archaebacteria, Dinoflagellates, slime mold. Methanogens belong to Archaebacteria. Slime molds are, to which kingdom do they belong? Slime molds? Protista. Slime molds belong to Protista. Dinoflagellates? Dinoflagellates? They are also protistans. Select the wrong statement. The walls of diatoms are easily destructible. True or false? False. Why? Diatom wall has silica crystals, granules. So diatoms are also referred to as siliceous earth, silica earth. Not easily destructible. Diatoms are the purest form of silica available. Diatoms are the purest form of silica available. Okay, the other things we'll see. Diatomaceous earth is formed by the cell wall of diatoms. True. Diatoms are chief producers in the ocean. Yes. Diatoms are microscopic and float passively in the water. That is also correct. Diatoms are a part of the phytoplankton population. The label of a herbarium sheet does not carry information on. What information does the herbarium sheet not carry? Date of collection, name of the collector, local names, height of the plant. Height of the plant is irrelevant information because if you take a young plant it will be shorter, if you take an older plant it will be longer. It does not add any value to the information. But the rest of the things are important. Date of collection, you know at which season you have collected it and it's a reference point. Doesn't change with whoever does it. Name of the collector is important because any cross-referencing you have to do, any information you need about it, you need to know who to reach out to. Local names, important. Height of the plant is insignificant. Conifers are adapted to tolerate extreme environmental conditions because of. What is special about conifers? Broad, heavy, hardy leaves. Superficial stomata, thick cuticle, presence of vessels. Cuticle is composed of what? Cuticle. Thick cuticle, if they have thick cuticle, what is the purpose? What is the risk for conifers? Where do they grow? They grow in cold and dry climates. So why do we need a thick cuticle if they are growing in cold and dry climates? Why do we need a thick cuticle? What is the purpose of cuticle? What does it help the plant in? Cuticle helps the plant to reduce water loss. Cuticle is made up of wax. Waxy cuticle prevents water loss. But if it is already growing in a cold region, why do you need to prevent water loss? Extremely cold regions, there is no transpiration loss, but the water availability is also low only, no? Because water is in the form of ice. Crystallized water is not available for plants to utilize. So they also have to operate in a minimal water availability conditions. And what is wrong in statement in first option, broad hardy leaves? What type of leaves do they have? Conifers? Needle-like leaves. Superficial stomata? Do they have superficial stomata? What do they have? Sunken stomata. Sunken stomata, not superficial stomata. Presence of vessels? Conifers are gymnosperms, so they don't have vessels. The xylem of gymnosperms lack tracheids. Lack trachea, they have only tracheids. Which one of the following statements is wrong? Algae increase the level of dissolved oxygen in the immediate environment. Is it correct or wrong? It is correct. Why algae are photosynthetic? So they add more oxygen, increases dissolved oxygen because they are in an aquatic environment. Argin is obtained from red algae and carrageenan from brown algae. Same question in the previous paper also. Algin is obtained from brown algae and carrageenan is obtained from red algae. That is the wrong statement. Agar agar is obtained from Gelidium and Gracilaria. That is correct. Laminaria and sargassum are used as food. That is also correct. Same question only, no? The question also was the same. Question and options. Our options are changed. I think in the previous paper it was chlorella as the food source. One of the option was chlorella, not laminaria and sargassum. The term polyadelphous is related to Gynoecium, Androecium, corolla, calyx. Polyadelphous, Androecium, fused stamens, multiple stamens fused to form a single bundle is polyadelphous. No, not fused, separate. They are separate. Stamens are separate is polyadelphous. If they are all fused to form one bundle, what do you call them? What do you call?
Monoadelphous.
Monoadelphous. So you have more than two bundles, then it is polyadelphous. Fused gynoecium is called syncarpus. Corolla and calyx are non-reproductive whorls. This term does not refer to them. How many plants? How many plants among Indigofera, Sesbania, Salvia, Allium, aloe, mustard, groundnut, radish, gram and turnip have stamens with different lengths in their flowers? Next question is. No, my sincere advice is you should not even bother to prepare for such questions. If at all, such questions will not appear anymore. It has become more sensible. The questions have become more sensible, more logical, where you have to apply logic and analyze. How on earth can you remember? Unless you memorize a list of common plants and what kind of, and you have gynoecium itself, superior ovary, inferior ovary. Forget it. Radial symmetry is found in the flowers of Brassica, Trifolium, Pisum, Cassia. Radial symmetry is actinomorphic. Radial symmetry is actinomorphic. Brassica is a the group that we studied. Brassica is actinomorphic. Okay. Trifolium, Pisum, Cassia. Do we still have this in the syllabus? Unfortunately, we do. Okay. Free central placentation is found in. Oh, this paper is full of this kind of questions only. Free central placentation is found in. What do you mean by free central placentation? What is free central placentation? Placental axis is in the center and the ovules are all attached to the central placenta. There is no connection with the ovary wall. Ovary wall is not, seen. Dianthus is free central placentation. Argemone, parietal placentation. You can remember Brassica, parietal placentation. Citrus, axial placentation. Can you recall the different types of placentation?
Yes.
Tell me.
Marginal.
Marginal, axial, basal, superficial, parietal, then free central. Okay. Cortex is the region found between epidermis and stele, pericycle and endodermis, endodermis and pith, endodermis and vascular bundle. See, as you move from outside to inside, what all do you encounter in any plant tissue? The outermost covering is the epidermis. Then as you move inside, you get the cortex. Some of them might have a hypodermis. Sclerenchymatous hypodermis is found in the stem of monocots. Dicots don't have hypodermis. You have cortex. After cortex, you have the boundary of cortex, endodermis. Endodermis. After that, you have pericycle. As you move inside, the entire medulla In the medulla, all the vascular bundles are referred to as stele. Center of the medulla, core of the medulla is the pith. Core of the medulla is the pith. If you know the relative position of different structures, you can answer this. So cortex is the region found between what? You think about it now in the sequence. Between epidermis and endodermis. Between epidermis and endodermis. That is not given. So what do we look at now here? Epidermis and stele. Epidermis and stele. The vascular tissue is referred to as stele. Balloon-shaped structures called tyloses. Where do we find tyloses? In which tissue do we find tyloses? Tyloses are extensions of xylem parenchyma into the vessels. Balloon-shaped projection of the xylem parenchyma cells into the vessels. So what will happen if these cells bulge out into vessels? Xylem vessels or trachea? It will block transportation in that particular vessel. Tyloses will block transportation in that particular vessel. Okay, where do the balloon-shaped structures called tyloses originate in the lumen of vessels, characterize the sapwood, are extensions of xylem parenchyma cells into vessels, are linked to the process of guttation. What is guttation? Guttation happens where?
Margin of the leaves. The margin.
Tip of the leaves. That is the end of the xylem in the leaves. That is the terminal part of the xylem in the leaves. Guttation is caused due to what? Guttation is a proof of what?
Transpiration.
No.
Root pressure.
Root pressure. Guttation is the proof for root pressure theory. So that dewdrop that you find at the tip of the leaves in the morning, early morning, that is the fluid that is coming out of guttation. That is guttation. Alright. So balloon-shaped structures called tyloses are extensions of xylem parenchyma cells into the vessels. They are characteristic of heartwood and not sapwood. Okay, non-proteinaceous enzyme is, this again it's a repeated question. Lysozyme, ribozyme, ligase, deoxyribonuclease. Ribozyme. Ribozyme is a RNA enzyme. Name the enzyme, name one ribozyme. Where do you find ribozyme? Tell me the specific name of the ribozyme enzyme that you have studied.
23 sRNA in bacteria.
What is the enzyme?
Peptidyl transferase.
Peptidyl transferase is a ribozyme. Peptidyl transferase is the enzyme which will bring about transfer of amino acid from tRNA moving from the what side to what side? P side to E side and shifting into the tRNA coming into the P side. What is the function of ligase? Any ligase enzyme does what? Joins. Molecular glue. Ligases are referred to as molecular glue. What enzymes are referred to as molecular scissors? Restriction endonucleases. Restriction endonucleases. Select the mismatch. Gas vacuoles, green bacteria. Gas vacuoles, are they found in green bacteria? Gas vacuoles are found in cyanobacteria. Cyanobacteria have gas vacuoles. Large central vacuoles. Cyanobacteria is also blue-green algae, green bacteria, you can consider that, photosynthetic bacteria, that is correct only. Large central vacuoles, animal cells, is it right? Plant cell. So you don't have to look at anything else, that is the by default wrong choice. Protists, eukaryotes, methanogens, prokaryotes, all others are correct except the second one. Select the wrong statement. Bacterial cell wall is made up of peptidoglycan. Is it wrong statement? No. Pili and fimbria are mainly involved in motility of bacterial cells. Is it wrong statement?
Yes.
Yes. What are they involved in? They are involved in attachment, adhesion. Pilus and fimbriae help in attachment of the bacteria to the surface and within the colonies to each other. Cyanobacteria lack flagellated cells. Cyanobacteria don't have flagella, they are aflagellated cells. Mycoplasma is a wall-less microorganism. It is correct. It is a correct. It is a correct statement. What is it? Mycoplasma. We are telling it is a microorganism, but what is it? What microorganism is it? Bacteria. Mycoplasma are bacteria. So only bacteria, which the known bacteria that does not have a wall. A cell organelle containing hydrolytic enzymes. Easy. Which organelle has hydrolytic enzymes? Lysosomes. Lysosomes. What are microsomes? Fragments of the endoplasmic reticulum. Endoplasmic reticulum breaks off and you find fragments of the endoplasmic reticulum, they are microsomes. So microsomes, if they are fragments of rough endoplasmic reticulum, can do protein synthesis also. Ribosomes, we know involved in protein synthesis, they don't have any hydrolytic enzymes. Mesosomes, invaginations of the bacterial cell membrane. What all can mesosomes do? They can do photosynthesis in photosynthetic bacteria. They can do respiration, functioning as a temporary mitochondria. Alright? They are like temporary aggregates. It is like tent. Organelles are like permanent house. Mesosome is like a tent. Whenever you want a shelter for people, a set of people, you put a tent. That is mesosome, temporary functioning organelle. During cell growth, the DNA synthesis takes place in. We have already discussed this too many times. During what phase? S phase. Which of the following biomolecules is common to respiration mediated breakdown of fats, carbohydrates and proteins? Repeat question. What is common to all three pathways? Acetyl-CoA. Acetyl-CoA. See, repetition already. A few drops of sap were collected by cutting across a plant stem by a suitable method. The sap was tested chemically. Which one of the following test results indicates that it is a phloem sap? Which one of the following test results indicate that it is a phloem sap? Phloem sap contains what? If it is phloem sap, what should it contain? What should it contain? Sugar. If it contains sugar or sucrose and other organic content, will it be acidic or alkaline? It is alkaline, it is not acidic. So acidic is wrong, alkaline is right. Low refractive index. Will more sugars in a particular fluid reduce its refractive index or increase its refractive index? Refraction is the ability of a substance to bend light, reflect light. So you have this, no? What is that property called? Sugar. What is the property of sugar where it deviates light? Come on, tell me. Correct.
Inversion.
L and D. Levo and dextro rotation.
Yes, sir.
Rotation of polarized light, no?
We say it's an invert sugar, sir.
Invert sugars. Fructose is? Is fructose an invert sugar? It is it. So refractive index increases. Absence of sugar is wrong because phloem sap contains sugar. It has more sugar. You are given a tissue with its potential for differentiation in an artificial culture. What do you call that? If a tissue has potential for differentiation in an artificial culture, what do you call that mass of tissue? in tissue culture terms? What do you call it? Not stem cell. What do you call an undifferentiated mass of tissue produced out of tissue culture technique which has the potential to differentiate under the influence of specific hormones? Callus. Callus. So here the question is talking about something maybe related to callus. We will see. You are given a tissue with its potential for differentiation in an artificial culture. Which of the following pairs of hormones would you add to the medium to secure shoots as well as roots? IAA is auxin, indole acetic acid, auxin and gibberellins, auxin and cytokinin, auxin and abscisic acid, gibberellin and abscisic acid. What are the two hormones you need to add for bringing about shoot differentiation and root differentiation? Auxin and cytokinin. Adding which one in higher concentration will bring about shoot differentiation? More cytokinin, shoot differentiation. More auxin, root differentiation. More auxin, root differentiation. More cytokinin, shoot differentiation. Should remember this with tissue culture. You need to remember callus and this. Phytochrome is a flavoprotein, glycoprotein, lipoprotein, chromoprotein. Flavoproteins are those proteins with flavin as a cofactor. Okay, glycoprotein is proteins with carbohydrate groups. Lipoprotein is proteins with lipid groups. Chromoprotein is pigmented, color for producing protein, pigmented protein. Phytochrome. Phytochrome is what? It is a chromoprotein. It is a photoreceptor pigment. Phytochrome is a photoreceptor pigment which regulates process like seed germination and flowering in response to light. How does the plant identify the presence of light and response? Phytochrome contributes to that. It is a chromoprotein. A protein which is absorbed, which is bound to a light absorbing component. Which one is essential for the growth of root tip? Mineral nutrition. Out of syllabus. Calcium. Process which makes major difference between C3 and C4 plant is photorespiration. Photorespiration. C4 plants have a specialized enzyme. What is that specialized enzyme which will convert C3 compound into a C4 compound? PEP carboxylase. Phosphoenol pyruvate carboxylase. PEP carboxylase is the enzyme. Where do you find this C4 pathway happening? In the bundle sheath cells. Bundle sheath cells. Which one of the following statements is not correct? Offspring produced by the asexual reproduction are called clone. Is it correct? Yes. Asexual reproduction, offspring is clone. You pick up any somatic cell, stimulate it to develop into an embryo, and that embryo develops into the offspring. It is a clone, exact same DNA, exact same chromosome, that is clone. Microscopic motile asexual reproductive structures are called zoospores. Microscopic, yes, zoospores. Zoospores are asexual spores. And zoospores are motile. So that is also correct. In potato, banana and ginger, the plantlets arise from the internodes present in the modified stem. Is it right? Any stem giving rise to a new, any growth in a stem, whether it is an offset or whether it is a shoot branch or a leaf or a floral branch, whatever, or giving rise to new roots or a new plant itself, all of that will happen from nodes, not from internodes. Internodes are not associated with growth. So statement 3 is wrong. Water hyacinth growing in the standing water drains oxygen from water that leads to the death of fishes. Is it right? Yes. Water hyacinth is a kind of a aggressively growing plant species which grows very, it's a weed, it's an aquatic weed basically. Water hyacinth is an aquatic weed. High energy consumption, they not only energy depletion, they do one more thing which will destroy the other plants. What is it? They cover the entire surface, blocking sunlight penetration, light penetration into the water. They do both. Which one of the following generates new genetic combinations leading to variation? Which one? Sexual reproduction. Nucellar polyembryony means what? It is not nuclear polyembryony. Nucellar polyembryony. Take the cells of the nucleus and develop embryos. That will also give rise to identical embryos because all of them are coming from the somatic cells. Nucellus is a somatic tissue. Nucellus is a vegetative tissue. In plants we don't call somatic, we call it as vegetative tissue, a non-reproductive tissue. What is parthenogenesis? Offspring developing without fertilization, parthenogenesis, parthenocarpy. Vegetative reproduction, we know any vegetative part of the plant developing into a new plant. Matching question again. Catch hold of one thing that you are absolutely sure. Pistils fused together, syncarpus. Pistils or carpels or gynoecium, whatever it is, all of that is the same, syncarpus. So A should be 3. Done over. Only one option. Cross check with the other 3. B should be 1. Formation of gametes, gametogenesis. Hyphae of higher ascomycetes, dikaryotic. Unisexual female flower, pistillate. Done over. That's all. What do you mean by dikaryotic? Karyon means what? Nucleus. Dikaryotic means 2 nucleus. In majority of angiosperms, egg has a filiform apparatus. There are numerous antipodal cells. Reduction division occurs in the megaspore mother cells. A small central cell is present in the embryo sac. Now, does egg has filiform apparatus? What has filiform apparatus? Synergids have filiform apparatus. So, first statement is wrong.